Okay, so Bharadwaj will talk about evolutionary paradigm favoring cross between bacterial to component signaling systems. Go ahead, Bharadwaj.
Hello. Yeah. Hello. Every organism, simple or complex, needs to sense its environment and respond accordingly. This task of sensing and responding to stimuli in bacteria is primarily mediated by what are called two-component signaling systems, or TCSs in short. Let me explain how they function. This is the cross section of a bacterial cell. The first component of a TCS is a transmembrane protein called the histidine kinase HK. Upon sensing a stimulus, which is typically a chemical ligand, it takes up a phosphoryl group and becomes active. This active HK then transfers its phosphoryl group to the second component of this TCS called the response regulator RR. The then active RR triggers downstream gene expression and elicits the required response. This response is twofold. One, it of course produces the proteins that are required to elicit the response, and the second feature of this response is upregulation of the TCS system which got triggered, basically making more and more of these HKs and RRs. Different TCSs are required to sense different signals, and here I'm distinguishing them using different colors. And now the question is, how does a bacterium evolve distinct TCSs, and what are how the evolutionary forces shape them? TCS proteins, which are HKs and RRs, share significant homology, meaning they are similar to each other. This is because a bacterium acquires new TCSs through a process called gene duplication. In this process, the gene which encodes for the TCS duplicates, of course, and then a process called domain shuffling happens, which makes sure that the newly arised TCS would be able to detect the signal which the parent TCS cannot. This is when crosstalk could be preserved. Here I define crosstalk as the possibility of the HK of a TCS system to transfer its phosphoryl group to the RR of a completely different TCS system. In this cartoon here, I'm showing crosstalk using red dashed arrows. It can be present in either ways, although I'm showing it here in a bidirectional fashion. There are a few obvious disadvantages of having crosstalk. Of course, the signal gets dissipated, weakening the response, and also unwanted responses might get triggered. Thus, along these lines, there have been many experimental and bioinformatics studies which have proven that there is strong evolutionary force for these TCSs to be specific, or in other words, insulated from each other. I'm listing here several bacteria, a few bacteria which are widely studied in this field, the number of TCS proteins that their genomes are known to encode, and the number of the percentage of crosstalk interactions of all the experimentally known interactions. Of course, there is a large chunk of bacteria which do follow the specificity paradigm, but there are bacteria, especially like the Mycobacterium tuberculosis, the pathogen for tuberculosis disease in humans, which is known to have a lot of crosstalk interactions. Now, it is important to understand what are the evolutionary forces that have shaped for the selection of crosstalk in such bacterium, because they might have implications in understanding their adaptation mechanisms, and thus possibly in designing future therapies. Hence, we set out to test a hypothesis, which is crosstalk between these TCSs might have been an evolutionary outcome of the bacterium's programmed environment. By programmed, I mean that the environment elicits stimuli in a predefined sequence. To test this hypothesis, we have constructed a mathematical model, which I'll explain. Upon binding of the input signal, the HK gets activated. The activation status is shown here using a orange blob, the phosphoryl group. The then active HK then transfers its phosphoryl group to the inactive RR, turning it active. Also, it is known that inactive HKs acts upon active RRs to dephosphorylate the active response regulators. This is called phosphate activity. Finally, the response regulators trigger downstream gene expressions by binding to the promoter regions of the DNA. These histidine kinases can interact with non-cognate RRs at an attenuated rate gamma k, where gamma is the attenuation factor varying between 0 and 1, and K is the strength of the cognate interaction. So basically, the crosstalk interaction is weaker than the cognate interaction. Finally, since a bacterium can have tens of TCSs, and since depending on the presence or absence of crosstalk interactions, there are multiple phenotypes that are possible. So we have identified each of these phenotypes using what we have devised as an interaction matrix. An example is shown here for a bacterium with 4 TCSs. The orange colored diagonal elements represent the presence of the cognate interaction, which are always there. The blue colored off diagonal entities represent the presence of a crosstalk interaction, and if they are white, they are absent. And the rate at which crosstalk happens is at the attenuated rate gamma. There are many such phenotypes that are possible. Now, to quantitatively compare the performance of each of these TCSs, we have constructed a fitness metric phi I of t for every ith TCS in a bacterium. It consists of 2 elements. The magenta element is a negative of the normalized input signal, which makes sure that negative of the normalized input signal, because higher the input signal, higher is the requirement for adaptation, so lower will be the fitness. And the red term here is the fraction of free promoters, which is 1 minus the fraction of bound promoters. So higher is the fraction of bound promoters, lower will be the fraction of free promoters, so higher will be the fitness. The construction makes sure that the fitness varies between 0 and 1, and for every bacterium with n such TCSs, for example, the fitness of a bacterium is the product of the fitness contribution from each of those TCSs. Finally, we have tested our model in 2 environments. The first is, of course, programmed. We have stuck to the signal sequence, signal 1 followed by signal 2 followed by 3 and so on till n. Any such sequence can be used. In the random environment case, all the signals have equal possibility of coming up. Now let me explain the results for the simplest case possible, which is a bacterium with 2 TCSs. There are 4 phenotypes that are possible. The first, phenotype 1 without any crosstalk, the phenotype 2 with crosstalk between HK1 and RR2, the phenotype 3 with crosstalk between HK2 and RR1, phenotype 4 with bidirectional crosstalk. And I have depicted the respective interaction matrices on the bottom. So when we simulate this model, we have simulated this model in a programmed environment, and that's what these results are. In the programmed environment, as I've said, the signal 1 is followed by signal 2, and the signals come in a step fashion, 1 followed by 2, and both the signals stay for a equal duration of 500 seconds. The panel on the top plots the active RR concentration in each of these phenotypes, and the panel on the bottom plots the fitness of each of the TCSs in the bacterium. And the curves are color coded for each of those phenotypes, where the dark and the light curves correspond to the TCS1 and the TCS2. For phenotype 1, where the TCSs are insulated, the response you can see here from the black and the gray curves are identical except for the shift in time. This was expected because both the TCSs are insulated. But when you look at phenotype 2, which is in red, the TCS1's response when signal 1 is present is weaker relatively compared to the phenotype 1. This is because there is a small amount of signal that is getting dissipated from HK1 to RR2. But upon the onset of signal 2 in the environment, we see that the light red curve is much above the gray curve for a significant amount of time, meaning that the organism is able to respond much better compared to the TCS2 is able to respond much better compared to the TCS2 of the phenotype 1. This has been reflected in the fitness curves as well. This is because when the environment has elicited signal 1, there is a small amount of signal that got dissipated to TCS2, which has upregulated the TCS2 in the phenotype 2, basically sensitizing the bacterium for the upcoming signal. So as and when the I2 signal has been elicited by the environment, the phenotype 2, which was already sensitized, was able to respond much faster. Phenotypes 3 and 4 actually lose out in this fitness game because there is unnecessary phosphatase activity by HK2 when I1 is present, and also there is unnecessary dissipation of signal when I2 is present. So in short, when you see at the net fitness cost of the organism 1 minus phi, in this whole simulation, you see that the phenotype 2 pays the lowest amount of fitness cost to adapt to these changes. Now this whole analysis was done for a specific value of gamma, but we observe that even though we vary the values of gamma, we see that the selection coefficient, which is defined as the relative fitness of each of these phenotypes compared to phenotype 1, wild type phenotype, we see that the phenotype 2 has the highest selection coefficient or positive fitness. But as one might right now expect, the fitness does not stand. The fitness advantage should not stand over all values of gamma, and so that is what we see. This is because the advantage because of priming saturates after some time, but the cost that is paid by the TCS1 because of dissipation of signal continuously increases. But at the same time, if we redo the analysis for the random environment case, we see that any crosstalk is detrimental, and as value of gamma increases, the cost that is paid by the organism also increases. Hence, crosstalk mirroring the signal sequence could be advantageous in the programmed environment. So finally, we set out to see whether this fitness advantage is significant enough for for the bacterium to be selected in the population. We have done in both the random and the programmed environment cases with two different initial conditions. And what we see here is that the blue curves in the random environment case, which correspond to the phenotype without any crosstalk, dominates over the population, and the red curves in the programmed environment case, which has crosstalk mirroring the signal sequence, dominates in the population, and even the rank ordering of the phenotypes as per the fitness values also shows the same. Finally, we set out to see if there are evidences that can be found out from experimental data. So we try to look at the charted out data from M. tuberculosis bacterium, where they have charted out the network. The left column is of the HKs, the right column is of the RRs. The green arrows are the cognate interactions. Red dashed arrows are the experimentally observed non-cognate interactions. When we draw out cascades from this network, we see that there are multiple sizes of these crosstalk interactions that can be drawn out. And if we superimpose these networks on the top of the other, we see that there are many such crosstalk interactions that can be drawn out. Hence, we see that in conclusion, we see that these experimental data also lend support to our hypothesis. So to conclude, we have devised a mathematical framework to analyze crosstalk in bacterial TCSs, and we were able to present an alternative evolutionary paradigm where we find that the crosstalk mirroring the signal sequences could provide a fitness advantage in programmed environments, while it does not. It is of no use basically in random environments. Hence, we have tried to reconcile the prevalent specificity arguments with our crosstalk arguments, and we also try to provide evidences for the same from experimental data. Thank you.
Thank you for that great talk. We have time for 2 questions. Is there any question?
Yeah, so is there a relation between the number of HKRRs that a bacterium has and the crosstalk between these different HKRRs?
So experimentally, there is no relation as such because it depends on the stimulus that the HK detects. So in our complete model, we have completely generalized it. So from our model, you see that for a bacterium with n TCSs, the number of crosstalk patterns that are possible are 2 to the power n times n minus 1. So even though it is 4 for a case of n equal to 2, for a case of n equal to 4, you can see ideally 4,000 phenotypes are possible. So it blows up. So we don't really know whether there is a correlation between them or not, because people do not really chart out the stimuli that are trigger these TCSs. So we can't pre-guess the crosstalk pattern.
Why I ask is because I noticed in the initial slide that the M. tuberculosis has the lowest number of your HKRRs and
Yes.
it has the highest number of crosstalk.
Correct.
So it might just be that to like using lesser number of
Yes.
HKRRs, it is doing many things by doing the crosstalk in a combinatorial fashion.
Correct.
It's increased.
So basically, it is compensating for the lack of TCSs.
Number, yeah.
So we try to pursue in that direction as well. So confusingly, the Mycobacterium family has very less number of TCSs. So the avium Mycobacterium avium species has only 4 TCSs, but it still causes tuberculosis in avian species, and there is no known crosstalk as such. So there is no real correlation that can be drawn on between the number of TCSs and the number of crosstalk interactions.
Okay.
Yeah.
Thank you. Thank you.
Yeah.
So that is the end of this session. I thank all the speakers and thank you all for staying back and asking great questions. So thank you.